We're taking a look at Unit 3, Lecture 5. In this lecture, we start looking at the third line of the immune system. And in this lecture, we're going to be taking a look at lymphocyte development and the characteristics of antigens. So, the third line of defense is one of the more important aspects of your overall immune function. And where this becomes critical, as it says here, is when the first and second lines fail to prevent a pathogen from establishing in the body, meaning that the pathogen has come in, the immune system in the innate stage has failed to remove it, and now the pathogen is proliferating and potentially spreading throughout the body. So while the innate immune system is very often effective, this third line is necessary when the second line fails. Now, the innate line will typically remove a large number of microbes that come in, but at some point, and really it's just a matter of time, the innate system will fail at some point, and at that particular situation, the third line of defense will become critical. And ultimately, its ability to function is a matter of life and death. So you can go a certain period of time relying largely on the first and second line, but at some point, you'll have a microbe breach and it will require your third level of defense. So there's a few extreme examples where you can see how serious this is. And this first one is called severe combined immunodeficiency syndrome. Big long acronym. This is called SCIDS. So it's a rare condition, but it kind of points to what I'm saying here being critical. This is a scenario with a rare genetic disorder, and it prevents your body from ever building the cells that are a key part of the third line. And these are B and T cells, which we'll get into here with this lecture and the next one. So with this disorder, you have no real ability to produce competent B and T cells, and therefore you don't really have any effective third line of immunity. And what ends up happening here is this usually occurs in young children, and they simply can't have interaction with any foreign microbes, or they will effectively die. And so this has been sometimes referred to as bubble boy syndrome, and in the past they've made an attempt to essentially isolate the child in a bubble from any contact with the outside world. And although that only works so well, that does prolong their life and keeps them from having exposure. So effectively, if they're exposed to any serious type of pathogen, they have no ability to fend it off and will die from that. So it kind of gives you an idea of without those cells, you are effectively just a sitting duck before any infection comes along and wipes you out. Another extreme example is AIDS, acquired immunodeficiency syndrome. This occurs when the HIV virus infects you and begins to replicate and spread, and typically it takes on average a few years before you develop AIDS. The virus kind of slowly cripples the immune system over a couple of years, and without treatment, the virus effectively wipes out the key cells in your third line of immunity. In this case, it happens to be a particular kind of T cell, but without that particular cell functioning, the whole third line begins to crumble, and you end up dying from any of a wide variety of infectious diseases that would otherwise not be a problem in the vast majority of cases. So it's another example of what can happen when the third line fails. AIDS is essentially a specific condition where the third line fails due to a viral infection in this case. So with that being said, most of us have a functional third line, and when it works, it typically kicks in after the first and second lines fail. Now, where the third line differs is it's referred to as adaptive, sometimes called acquired, kind of similar ways of describing the same thing. And then it can also be required specific. So as we get into this with this lecture and the next, you'll kind of understand why those terms are used. Can be a little confusing referring to the same thing with multiple terms, but all those terms kind of have their own context depending on how you're describing it. Now, this is without question the most complex part of your immune system, and that has to do with the specific interactions taking place. It also is sometimes referred to as a delayed response. because as the system is being activated, it typically requires several days, and in some cases a week or more, before the system fully begins to respond. And so for that reason, it also can be a problem in that even though it may be functioning, it doesn't start working immediately. The first and second lines are essentially rapid and quick responding, whereas the third line, especially with the first encounters of a microbe, is delayed. The two main cell lines that are involved here are the T cells and the B cells, technically T lymphocytes and B lymphocytes. So while there are other cells involved, these are the cells that we're going to focus on here. These cells end up acquiring huge amounts of resources in your body. You ultimately produce billions of lymphocytes. So at any given point in time, the average person will have literally billions of different variations of lymphocytes circulating all throughout your body, your lymphatic organs, and ultimately occupying lots of resources. It's one of the reasons why if you don't take care of your body, eat good food, sleep, all that, one of the first things that tends to fail is the immune system, or at least it starts to work less effectively. Now, B cells themselves will go on to produce antibodies. So antibodies are another key part of the third line of immunity. So you may have heard of antibodies. These are kind of a product of B cell production. The third line is usually activated by phagocytes, which are part of the innate immune system, really more of the second line. So it's not always the case, but generally speaking, what will happen is a second line cell, like a dendritic cell, will pick up on the presence of a microbe and then use the activation from the dendritic cell to go on and activate a T or B cell, which then turns on the third line of defense. And that's one of the reasons why there's a delay. It takes days, sometimes weeks, because that process of activating the dendritic cell then leads to the dendritic cell migrating and literally moving from one part of the body to another, and then activating the B and T cells, which then take a little bit of time to proliferate. They then have to migrate back to the site of infection, and that's one of the reasons why it takes a period of time before it actually becomes effective at removing the pathogen. So there's a lot going on in this line of defense, and that just kind of gives you a brief overview of kind of some of the things that we'll be talking about here. Now, to understand how the third line works, there's a few key concepts that you have to know. The first is that the third line responds to specific antigens. Sometimes these are called immunogens, and although these are technically different, these two terms are very often used interchangeably. So typically, unless someone's being very technical, the term antigen and immunogen are largely used interchangeably. And we'll come back to what those are in a little more detail here. Now, immunocompetence is ultimately your body's ability to respond to these antigens. In other words, it's essentially the ability to have an effective third line of immunity. So even though you may have a first and second line, you're not fully immunocompetent without a well-functioning third line. Specificity is this idea that lymphocytes target very, very specific regions of a foreign antigen. And not only are we looking at a particular molecule, but a particular region of the molecule called an epitope. And so I'll get into this here towards the end of this lecture to clarify kind of what I mean by that. But this is where we differentiate the second line. It can be referred to as non-specific, versus the third line, which is considered specific. So we're actually targeting individual molecules down to specific regions of a molecule. And that's kind of what we mean by specificity. So I'll clarify more on that here towards the end. Another key concept of the third line of immune system is that it has memory. And this is kind of where the term adaptive comes in. Memory is this idea that once we're exposed to an infection or a particular kind of antigen, our body builds a specific line of lymphocytes called memory cells. And these cells have a unique capacity to survive in the body for years, even decades, after the first infection has gone away. And this is what the idea of building immunity or building your immune system. You get sick, the body is kind of unable to respond effectively the first time, but after clearing the infection, you now have memory, and if the pathogen or antigen comes back a second or third time, with that memory in place, the body is able to respond to it much more effectively. And that's kind of the idea of building your immune system based upon memory. This is a figure out of your textbook that just gives you a few illustrations to kind of go along with these terms, and it adds a few more. So specificity means that we're reacting to very specific parts of a foreign entity. Diversity is the idea that we have lots of different types of B and T cells that each respond to different parts. And each cell is triggered to respond to a very particular piece of a microbe rather than just the microbe as a whole. Inducibility is the idea that we have to turn this system on. By default, it'll be turned off, and we have to turn it on typically by engaging with the second line or innate immunity. Once again, that takes time, so there's a bit of a delay there. Clonality is this idea that once we turn it on, we start to generate duplicate cells that all have the same specificity towards the relevant pathogen. So once we encounter a pathogen, we create clones or copies of cells that all respond to the same little antigen of that pathogen. Tolerance is this idea that we have to be able to differentiate between our own body cells and foreign entities, so that way we don't react to our own body. And memory, as I said, is this idea that once we engage a pathogen the first time, we remember it years and even decades later. Now, tolerance is important because the immune system is made up of cells that do not have a brain. So they have to be able to differentiate what is self and what is non-self at the molecular level. Now, honestly, this is one of the more complex parts of the immune system. So I'm going to lay out some concepts here, but I will not be able to go into the full level of detail. That's going to be way outside the reach of a class like this. But the way that it works is that you have a genetic inbuilt protein system that creates what are called major histocompatibility proteins, or what's generally referred to as the major histocompatibility complex, MHC. This can also be referred to as human leukocyte antigen, HLA. Science naming is always complex, and so you'll hear people refer to it in both different ways. They more or less mean the same thing. So these are genetically encoded, kind of like a fingerprint. And so the idea is that each person has a major histocompatibility complex, but it is unique to each individual person. The only exception is if you have an identical twin in which the zygote splits at the very beginning. So identical twins, not even fraternal twins, where you have two eggs fertilized simultaneously. So an identical twin would have the same MHC. Otherwise, every person in the world has a slightly unique MHC complex. So we all have it, but we all have a unique version of it. There are essentially two major classes of MHC, MHC1, class 1, and MHC2, class 2. So this is another area where it gets kind of nuanced. And again, with a class like this, we're not trying to spend too much time getting into the weeds. But MHC1 are going to be found on all cells with a nucleus, which really is all of your cells except for red blood cells. That's the major exception there. So pretty much all of your cells, other than red blood cells, will have this particular class. Class 2 are a little bit more specific. These tend to be found on phagocytic cells, more specifically macrophages, dendritic cells, and then B cells also have these. And these cells will also have class 1 as well, since they have a nucleus. So in other words, almost all cells in your body have class 1. Certain cells have class 2, these particular cells here. Now, what these allow us to do is to essentially determine whether the cells in our body are self, and it allows us to sample antigens from the body and present those antigens to T cells in particular that can determine whether a molecule is foreign or self, and therefore whether to attack it or to leave it alone. So it gives us the ability to determine self in a more complex way by displaying what is normal to cells and helping them determine what to attack and what to leave alone. Now, the way this works largely occurs during the development of the cell, and I will touch on that a little bit later here, but again, that's a nuance that we won't touch on every detail here. But this is a big, complex process happening as you're developing throughout your life, more heavily at a young age. And what's happening is that these cells in your body are taking proteins that are part of your normal body function, and they're displaying them on the surface of the cell and kind of telling the T cells what to think of as normal. And if we then have some kind of foreign pathogen, especially a virus in particular, viruses tend to infect cells internally. If a virus particle is picked up and displayed on the cell, the T cell will see that particle as being foreign and recognize that the cell is being infected. So you kind of have this very interesting process by which the cell is sampling and displaying and determining over time normal versus abnormal. So any protein that's not part of the cell that gets displayed is going to be recognized as foreign. Now, an interesting connection to cancer here is that when a cell becomes cancerous, some of the proteins start to become abnormal. That's one of the hallmark signs of cancer on a very technical level. And so cancer cells divide and become tumors. And in that process, what goes wrong is that the proteins themselves start to look abnormal. And so as the body displays abnormal cancer proteins, you actually have a mechanism by which cells in your body can recognize the cell as being cancerous and can actually eliminate it before it becomes a bigger spreading tumor or cancer cell. Okay? Now again, this concept is a lot more complicated than this few slides can possibly illustrate, but it gives you at least a little bit of an insight as to kind of how the body works. These MHC, major histocompatibility complexes, are sort of sampling and displaying, and through that process, the body learns what is normal and can then recognize what is abnormal. And so that's at least a sort of slight insight as to what's going on there. Now, the way that the immune system works is that it goes through 4 major stages of this third line. So this is really just in reference to the third line. So the first one really occurs throughout your entire life, and more heavily so in the early stages of your life. And this is called lymphocyte development. And the idea is that your body is just constantly churning out lymphocytes, largely from the bone marrow, and developing them into these billions of cells that occupy your body. And it never really ends. It's an ongoing process, but there's a more heavy development in the early years of your life. Now, once these cells are developed, they end up migrating into the secondary lymphoid organs. I talked about that in last week's lecture. And at that point, they kind of just sit and wait. And most of these cells will never be activated. But for those that are, what will happen is an antigen will be presented to them in one of a few different ways. And this is typically from dendritic cells, which are an innate cell. So if an antigen is presented to them, then they become activated. And at that point, the next stage occurs, which is said to be challenge. In other words, they've kind of been challenged to respond to the presence of an antigen. And at that point, the process kind of divides into 2 different stages, depending on whether you have a B cell or a T cell being challenged. Now, really, any given presentation will cause both to occur. Okay, so at that point, you start characterizing what happens in a B cell versus what happens in a T cell. So you kind of get this dual-pronged approach, one from the B cell line and the other from the T cell. And at that point, the cells are fully activated, and then they start to respond. So you kind of have ongoing development, periodic presentation, challenge is kind of the activation stage, and then lymphocyte response is when they actually become competent to respond to the presence of the pathogen. So B cells are, the response here is called a humoral response, and this is somewhat of an arbitrary way of describing it, but they call it a humoral response. And the main thing that B cells do is they produce antibodies. And antibodies then are really what go on to cause the more specific response in the B cell line. So ultimately, the B cells do their job by producing antibodies, and antibodies are essentially what remove the pathogen. T cells are a little more nuanced. They have what's called a cell-mediated response. And what they essentially do is attack cells that are abnormal. So these are, generally speaking, virally infected cells and or cancerous cells. And there's a few other ways that can work. But generally speaking, these are going to be cells that are abnormal, and therefore that typically implies that they're virally infected or cancer cells. And that largely goes back to that MHC response that I just showed there. And that's one of the ways that T cells kind of determine what cells to engage with. But the idea of a cell-mediated response is that they kill abnormal cells. They actually, these are going to be your own body cells. So if you have a viral infection inside of your cell, the T cell will come in and literally wipe out the virally infected cell. And then what your body has to do is replace it with a normal, non-infected cell. So there's a destructive aspect, self-targeting, that goes on in order to remove abnormal cells. So that's kind of a quick overview of the 4 stages. Development is a somewhat complex and ongoing process. So we're going to go through a few different aspects of development here. So these cells will begin in the bone marrow, and this is the process of hematopoiesis. I talked about that in the overview of the immune system. So hematopoiesis begins in the bone marrow, and you produce these lymphocyte cells. They're called lymphocyte stem cells, and they start to develop into the 2 different lines depending on kind of the specific process involved. Along that line, what will happen is each cell will start to develop its own little receptors that are kind of unique to each cell type. And these are proteins that the cell produces genetically. And depending on which cell line you're talking about, you'll produce a slight different variation of cell receptors. And that's really largely what gives rise to the difference between these 2 cells. Although there's more to it, it's the receptor configuration that tends to be the larger differentiator between the 2. Now, when B cells become a B cell, one of the things that happens is they develop almost entirely in the bone marrow. So those cells that are lymphocytes that develop entirely in the bone marrow get programmed within that environment to become a B cell. The cells that become T cells begin in the bone marrow, but then they end up migrating to a specific organ in the body called the thymus. And the thymus is an organ that has the specific function of developing T cells so that they become that specific cell line. And so while there's many other details, that's kind of the general idea, is that they start off as kind of a generic lymphocyte. They develop specifically into the 2 broad cell lines, and each one then gets kind of a unique configuration. From there then, they will migrate to the lymph nodes and end up waiting in the lymph node, which is technically a secondary organ, which they will sit there for long periods of time and wait for an antigen to be presented to them in a couple of different ways. So the T cells get their own unique receptors, the B cells get their own unique receptors, and then they move off into the lymph nodes waiting for an antigen to be presented to them. So going into a little bit more detail on that, the lymph nodes become then the housing spot for these, and the spleen is another area where they will sit. So these are kind of the 2 big ones, although there are others as well. And if you go back to the overview of immunity, I talked about a few others like associated lymphoid tissue, SALT, skin associated, M, mucoid associated, G, gastric associated. So there's a few other ones that they tend to go to as well. But once they're developed, they reside in these tissues here, these organs rather. And so they'll sit there until an antigen is presented to them in one of a few different ways. And at that point, what happens is the antigen binds to one of these receptors, and that binding activates the cell. And that begins this proliferation, which is kind of the initial stage of challenge. So the challenge, that third stage of the lymphocyte response, is kind of the what occurs after a lymphocyte has been presented with an antigen. So the antigen is typically brought in by a dendritic cell. It could also filter through the lymph node and kind of be brought in through the lymph fluid also, is another way. But when that happens, then the cell begins to go through what's called clonal expansion, which is a more technical way of describing this proliferation. So when we start off with these cells in development, as we'll see here on the next slide, we end up producing lots of different types of lymphocytes that all have the ability to respond with different receptors to different antigens. And that process is actually random, as we'll see here in a second. So we actually end up producing billions of random receptors that all have the ability to respond to different antigens. But once a particular cell has been activated by presentation, it will then go through expansion, clonal expansion. And that one receptor configuration that starts off random will then make copies of itself. Or the whole cell will make copies of itself. And each copy will have the same antigen receptor. And so what will happen then is once you have a particular antigen that activates it, you'll now copy that cell, and you'll now have lots and lots and lots of copies of just that one receptor configuration. And this is the idea of adaptability, adaptive immune response. We end up creating copies of the cells that we need based upon the antigens that are presented. And so that's the way that we kind of adapt or acquire immunity. We actually start off with lots of random receptor configurations that we never really use, but then we copy the ones that we do use or that are activated, and we end up with duplicates of the ones that are useful. And those end up staying in the body for long periods of time due to the memory that I talked about earlier. Now, the way that we get the receptor that the antigen binds to is a tricky process. But ultimately what happens is this starts out at the genetic level. And in our DNA, we have about 500 different genes that each contribute proteins into the building of these antigen receptors. So this figure here attempts to kind of show what that looks like. In the DNA, we have lots of different genes in different segments of the DNA. And each of these colors represents these different gene segments. So different coding regions represented by different colors. Now, if you remember how the DNA expression works, or gene expression, each gene produces a protein. And in this case, it's a smaller fragment type protein. And it's almost like a little Lego block. And what happens then is each individual fragment is then combined with more fragments, and these fragments then collectively become part of the receptor on the B and T cells respectively. So this Y shape here kind of represents the entire receptor, and each little tip of the receptor is put together by individual little protein fragments, each fragment coming from a different gene, and the combination of those genes building out the receptor in its entire configuration. And this process is called VDJ recombination. Ultimately, what this does is it kind of randomly scrambles these genes, all of which kind of have specific configurations. But every time a receptor is built, the receptor is built from kind of a random combination of different fragments, kind of like taking different Lego blocks and assembling them in different combinations. And what that does is each receptor then ends up with a unique configuration so that it can bind to any almost infinite variety of antigens. So what the body does is it builds a wide variety of receptors in anticipation of almost any antigen that you could be encountered with at some point in the future. And what this actually does is it allows us to respond to an antigen that doesn't even yet exist. We could have a new foreign microbe that has yet to even exist, and we could have a receptor that could bind to it even though we've never encountered it before. So it's kind of confusing, but ultimately it gives us the ability to respond to a near infinite array of antigens. And this kind of gives you an idea of how powerful the third line of the immune system is. Where the second line is quite different. The second line has similar kind of receptor configurations, but they're already preconceived. In other words, those receptors are predetermined to respond to things that our body has already encountered, specific configurations on bacteria and viruses and things like that. With this configuration, you build receptors before you've ever even encountered anything, and you build literally billions of them. So you have billions of different receptors that could theoretically respond to anything, even though you've yet to encounter it. But only when a particular unique configuration is activated will that particular version then be useful. That's when the body copies it to make many duplicates of that useful version, and then that version goes into effect to respond to the pathogen. It's a very confusing concept. This figure here shows it a little differently. Each of these are the VDJ segments. That's kind of the variable region that is unique. And so these kind of represent them in different box sizes and colors. And the combination of these then puts together different segments like Lego blocks to make a unique configuration that is then uploaded into the tip of the receptor, which is where the antigen will bind. So at that tip of the receptor, these different combinations are mixed up. You get a unique little configuration at the tip there, and that is ultimately going to be responsible for binding the antigen that could come in any theoretical infinite variety of shapes and sizes. So hopefully that makes sense. If it doesn't, I understand, because that is a confusing concept. But ultimately the idea there is that you put together a near infinite number of receptor combinations by using different gene combinations and different protein combinations. Now, one of the problems with doing it this way is that you will, for sure, put together a receptor that can target your own body. Since these receptors are randomly assembled, they could theoretically respond to any kind of organic material, including your body's own cells. Now, this is anticipated, so the body has a way of dealing with this. But this is one of the problems that arises with this mechanism. And since these antigens are relative, and our lymphocytes are kind of quote-unquote mindless little cells, they don't necessarily have a brain to differentiate what's good and what's bad, what's self and what's foreign. And so if you were to just build these cells and then release them all into the body, you would actually generate autoimmunity. The body would attack itself. So some of those receptors would for sure target your own body cells just by chance, and that would lead to autoimmunity. Now, the way the body deals with this is as it builds these random receptors, it then actually self-selects only those that don't bind to our own body tissue. And this is another level of complexity of the immune system. But the idea is that as we build these receptors, we essentially remove any of them that target our own body tissues. And this is another sort of complex part of the development process. So what you effectively do is build the receptors in a random way and then test them out. And you test them out to see early on, are they going to respond to your own body tissue? And if they do respond to your own body tissue, then the body has a way of removing them before it releases them into the broader circulation of your body. So this is actually taking place in a little specific area, largely in the thymus. So the thymus is a big area where this takes place. And the process is called clonal deletion. So any clone, so to speak, of a lymphocyte that does target the body's own tissue is removed very early in the development process. So you're creating random configurations and then you're testing them. And if they react to your body's own tissue, you remove them. And therefore they never really make it through the whole development or maturation process. And done correctly, This leads to what's called immune tolerance, the ability for the body not to target itself. Now, if that does not work properly, then you could end up releasing cells into the body that target your own tissue, and that could lead to autoimmune diseases. So some of these autoimmune diseases are thought to arise when this process fails. As I say here, it's important to note that there are other ways that autoimmunity occurs. And so while this is one way, failure to remove the self-targeting cell, there are actually many other ways that autoimmunity occurs in addition to this. But that's kind of an insight to the complexity here. Build a bunch of random receptors, and that gives you the infinite ability to respond to pathogens that may not even exist yet, which is important. But then the problem being you can target your own self, so the solution is to remove the self-targeting cells before they finish development. If that works perfectly, you will not have autoimmune diseases. If there's a defect and you get self-targeting cells that are released, that could be one contributing factor towards autoimmune diseases. So I hope that makes sense. I certainly understand if that's confusing, because the first time you hear this, you're kind of thinking to yourself, what? So you may have to go back into the textbook, review that, rewind this video, watch it again. But that's my best attempt to explain that in a relatively short period of time there. It's a very difficult thing to fully understand. But this figure does a little bit better job of showing it. So lymphocytes develop as stem cells. You create random receptor configurations, represented here by these little tips on the cell. And then early on, anything that responds to the self is eliminated through clonal deletion. So therefore, only the cells that don't respond to the self make it through development here. So these kind of represent here in the middle all the cells that actually make it through development from beginning to end, so that they don't target the self. Now, these cells will ultimately migrate out into the body tissue, into the secondary organs like lymph nodes, and they'll just sit there and wait. And most of these cells will never be activated. Most of them will end up having a short couple month duration, and then they'll eventually die, be broken down, and new cells going through the same development will replace them. So every couple months, these cells die and have to be replaced by a new cell. Very energy intensive, by the way. Your body consumes lots of new resources to keep this process going. Now, for those cells that are activated, what will happen is an antigen, or an epitope as it describes here, is bound to a specific receptor. So in this figure they're showing of all the receptors that you build, this one actually encounters an antigen. And now what happens is this cell gets a signal that says you need to reproduce or go through clonal expansion. And so now that one cell makes a copy of itself over and over and over, thousands and thousands of copies. And now you get lots of duplicate versions of just that one configuration. And now these are all the same. They started out random, but now they all have the same configuration, and they will all respond to the same antigen or epitope that was initially encountered here. So this is the idea of adaptability. You have the ability to respond to virtually anything, and then you make copies of those that you do respond to, and your body gets more efficient at responding to the particular types of foreign entities that you're actually exposed to. That's the idea of building your immune system. So going on a little bit further here, the idea is that this process of copying the relevant receptor configurations is clonal selection and expansion. So the lymphocytes are released. They end up having a match to their specific receptor. They are therefore said to be clonally selected, meaning that particular receptor has found a match. You now copy the lymphocyte that was matched in order to make many, many, many versions, all with the same identical receptor. And those cells then become memory cells. I don't have that mentioned here, but the memory cells are those that last for long, long periods of time in the body. And that's how we kind of build long-term immunity, by creating copies that become memory cells that all match the same antigen and live for long periods of time. So most of the lymphocytes are not selected. They will effectively be removed after a certain number of months and replaced with new ones. Those that are matched tend to survive in the body for decades and sometimes longer. So that's the idea of building immunity. So to kind of summarize this, lymphocytes are created from stem cells with random binding receptors. They start off with any variety of receptor configuration. Any of those that bind to our own body tissue are removed very early in the development stage so that they should not be circulating throughout the body. Those that remain should only be able to respond to antigens or foreign antigens wherever they occur throughout the body. Only a certain number of those will actually be matched properly to an actual antigen that exists and is in the body. Those are therefore said to be clonally selected. The clonally selected lymphocytes will go through expansion, clonal expansion, making many, many thousands of copies of themselves. So from a random process to a non-self-selecting process to a refinement of the lymphocytes that actually encounter antigen. Most will not encounter antigen, and they will be removed and replaced by new cells of random configuration over time. So, as I mentioned, this process gives you the ability to respond to antigens that not only exist now, but could also exist in the future. And that's kind of the adaptability part. So once you once you respond to a particular antigen, you create this clone army with lots and lots of duplicates that can all respond to the same thing. So it begins randomly, but then focuses or hones in on what you encounter over time. It's an amazing system because it gives you the ability to change and adapt to things that could theoretically only exist in the future. Very much unlike the innate immune system, which can only respond to things that it's seen in the past. So while it is an amazing system, it's also very energy intensive. The billions of cells that you have to produce to make this system work require lots and lots of nutrients to keep that whole process going. And this is one of the reasons why if we don't take care of ourselves, we tend to get sick more often because we don't have the adaptive immune response functioning at the level that it should. As an example, vitamin D deficiencies are a major predictor of who's going to get sick and how likely they'll recover. So if you're deficient on vitamin D, which a large percentage of the U.S. population is, as a result, your developmental process and your ability to respond to pathogens in many different ways becomes hindered and less effective. And so when the body needs those resources most during an illness, you're going to be more deficient and unable to respond. And that could occur in the immediate sense or even over a long period of time. So just having proper nutrients of various different types of vitamins is a massive boost to the lymphocyte development process. Now, next here we're going to talk about what the body is responding to. So it's essentially responding to antigens. So these are the little foreign molecules that bind to the receptors, right? Those receptors are created randomly. We can respond to any wide variety of foreign entities, but what we're responding to is what we call an antigen. Now, to make a quote-unquote good antigen, it needs to have certain characteristics. Now, to say a good antigen is kind of like saying a good criminal. It's kind of like a backhanded compliment here. So, but in order for our body to respond to it, it needs to have certain characteristics. One of those is size. Smaller antigens tend not to be as effective. And the reason for this is that an antigen is actually made up of lots of different subregions called an epitope. So an epitope, as I'll show here on the next slide, is a specific portion of an antigen. So the terms are kind of confusing, but an antigen, as I'll show you on the next slide, is kind of the larger structure, and then the epitope is the particular piece of the structure that we respond to. Almost like if the car was an antigen, then the headlight would be an epitope, or the windshield wiper could be another epitope. So a single antigen, like a car, could actually have multiple epitopes. Everything from the headlight to the windshield wiper to the hubcap. And that's kind of the idea. Antigen, car, epitope, specific part of a car. And so larger molecules tend to be more antigenic because they have more little components that the body actually responds to. Now, in terms of organic matter, what our body is responding to, most of these antigens are made of protein. That is by far the most common kind of substance that the body responds to. Now, other times the body can respond to combinations, protein and lipid, fat, protein and nucleic acid, like DNA or RNA, protein and sugar, things of like carbohydrate. So those kind of combinations can also make antigens as well. And another class outside of proteins are the polysaccharides. These are large kind of sugar, carbohydrate kind of molecules. They're not quite as common to be antigenic, but they do act as antigens in certain situations. So those are kind of the general characteristics of antigens. Tend to be relatively large, lots of little subregions called epitopes, and they tend to be protein in nature in most cases. So when we look at the concept of an epitope, the idea, as I mentioned, is that the antigen is kind of the larger structure, and the epitope is a little specific component of the antigen, which is actually what our receptors and antibodies actually hone in on. These are also referred to as antigenic determinants as well. So large antigens will often have multiple epitopes, could be dozens or more, and the idea there is that we're honing in on a specific part. And in my notes here, I use the idea of a human face, kind of similar to a car. The face is a antigen, and the epitope might be something like the nose, eyebrows, chin, or lips. Just a different kind of analogy there. Very often what people say is that the body reacts to an antigen, usually just for simplicity. But actually, to be specific, we don't really react to the antigen. We react to the epitope, the little part of the antigen. So it's just kind of a matter of how specific you want to get, but technically speaking, we're reacting to an epitope, and that's why larger antigens tend to be more reactive or antigenic, because they have more components that the body can react to. So an antigen is a substance that we react to. Usually it's the antibodies, B cells, or T cells that are doing the reacting. The epitope is that specific component that we are reacting to. In some cases, what happens is we have a hapten. A hapten is a molecule that's too small by itself for the immune system to respond to it, but it becomes reactive when it binds to a larger carrier molecule. So a hapten is essentially an antigen that has to be attached to a larger structure. By itself, it's not antigenic. In combination with other carrier molecules, they become antigenic. So these are areas where the reactivity gets a little more nuanced. An alloantigen is an antigen that is present in some individuals but not others. So an example of this might be something like the A or B marker on your blood proteins or on your blood cells. So if I personally have a type A blood, and if you gave me type B blood, my body would see the type B as an antigen and attempt to attack it. A superantigen is an example of an antigen that tends to cause an excessive immune response. So rather than kind of a normal immune response, the presence of this particular kind causes an extreme immune response, usually resulting in lots and lots of inflammation and sometimes even death. Allergens are really antigens, but typically antigens that our body should not be responding to. And so what ends up happening here is the body responds to something that's harmless, but treats it as if it's harmful. And so the difference really between an allergen and an antigen is really just the nature of the substance or the protein usually. the body treats it like a harmful substance when in reality it's not. And this is the case with stuff like food allergens. If you have a peanut allergy, for example, your body responds to a peanut as if it's a foreign microbe, or dust, or pollen, any kind of allergy. The idea is that the body is kind of getting its signals mixed up and responding to something harmless as if it's harmful. So it's really just kind of the context there. The body perceives it as foreign, but doesn't necessarily need to, and gets the process kind of wrong and ends up causing more harm typically. And the last thing here to round this out is that the body becomes complex in terms of how it responds to an antigen in the sense of antigen context. So where our immune system starts to get really difficult to track is that the way it responds to an antigen is somewhat dependent upon the context. So for example, the cytokine interactions. So if you go back to what I talked about in the overview, I only mentioned these briefly, but cytokines are signaling molecules, and they basically communicate between immune cells like white blood cells. So depending on the kind of signaling that's going on, the body can respond to an antigen either more powerfully or less powerfully. In other words, more or less. So context here would mean what's going on with other cells around it. And this is kind of also related to inflammation. Inflammation is typically driven by cytokines. So if you have inflammation in the body, you're more likely to respond to an antigen than if you don't have inflammation. So having inflammation increases the reactivity to antigens that are present that otherwise might be left alone. Another nuanced part is the training that I've talked about. In the early years of your life as a young child, the body is sort of developing tolerance and kind of honing in on what to respond to and what not to respond to. And I mentioned how the microbiome is thought to play a role there, meaning the kinds of microbes that are present when you're first born train the immune system and kind of shape these responses so that some microbes and some antigens can be left alone depending on what you're exposed to at that time, and other types of microbes or antigens will be reacted to depending on what you're exposed to during that time. And this is why it's important to understand that children in the early years of their life are developing an immune system that will affect them for the rest of their life, whether good or bad. So exposure at that period of time is important, meaning that you don't necessarily want the wrong kinds of exposure, but you also don't want no exposure. So there are studies that show that kids who come from an environment that's too clean, for example, this is what's called the hygiene hypothesis. Kids that come from a home that's overly clean, it seems to underdevelop the immune system. They're not getting proper exposure. And then later their body ends up reacting to things that they otherwise wouldn't. So it seems to correlate with things like allergies and asthma and things of that nature, where because they weren't exposed enough when they were young, they end up overreacting when they get older. And so that's a hard one to fully characterize, but training during development is a key part. And the last thing here is that variability occurs by location. So the way that your body reacts in the lungs, for example, is different than the way it reacts in the gut, which is different from the way it reacts in the skin. And so what is going on in one body location, an antigen in the gut, for example, might be treated differently than an antigen in the skin, or vice versa. So the context of body location is another key thing there. So as I mentioned, these are sort of complicating factors that make it difficult to really track what the immune system is doing. But all of these things do influence the way the body responds to an antigen. All right, we'll wrap it up here with this one. And in the next one, we'll kind of take a more specific look at how B cells and T cells each respond to an antigen in the humoral B cell response and the cell-mediated T cell response in our next lecture.